research and clinical research, and so fortunate they have really dedicated their careers, their lives to the pursuit of better treatments and cures and current treatment of people living with ichthyosis. And we are so fortunate they're here with us this morning to talk about what's going on in research right now, what can we look forward to in the next couple of years. So I'd like to bring up Dr. Amy Paller and Dr. Keith Choate.
Well, first of all, hi, everybody. I hope you've had a great conference. I'm afraid Keith and I and our teams were buried a bit on 11. Thanks to those of you who came up and joined us for a while. It's kind of unusual to create a clinic in a hotel, but that's what we've been doing every 2 years, and thanks for putting up with that. What we're going to do today is Keith's going to talk a little bit about the registry, which, as you know, has been incredible as a way to drive forward research and our understanding, really through partnership with FIRST, with a lot of people coming engaged at this meeting and then in between. We and Keith have also done projects that we're going to talk to you a little bit about. Some of them small, low-hanging fruit like surveys, and others a little bit more extensive. I want to tell you a little bit about some studies coming up that we know about. And I know there's some here that I may not have the information for. So if any of you are in pharma and I don't have your information, make sure to get it to us, because we like to present this at regional meetings as well as at this more global meeting. So let's see what we've got. Okay, so you're coming up later then. Okay, good. All right, so a little bit about what's happening in clinical research, what's coming. This is just a little bit of highlights from some of the studies we've done in the past. I'm going to step forward a little bit so I can see. And I'm going to quickly go through these. But we did a study for ichthyosis looking at healthcare transition. This is always a problem for us as physicians. Many of us, for example, are pediatric dermatologists, and then there are adult dermatologists. And what happens is when patients go from the pediatric world to the adult world, there's a big transition. Now, I happen to be somebody who sees adults with ichthyosis because that's something I deeply care about. But that's unusual. And in many places, the pediatric dermatologist one starts with is not the person that will continue your care. Not to mention, you may start at a children's hospital, and then suddenly others who may be needed for your care don't see you. So there's always this concern about healthcare transition, and there are some scales that can actually be used to see how ready people are. So we did 45 participants with genetic skin disorders in this study, 31 of whom had ichthyosis, and what we found is that the readiness of the 14 to 22-year-olds was lower than for not just our controls, but for many other chronic medical problems, diabetes, sickle cell disease that have been out there. And just boiling it down to the main point, we found that transition readiness strongly correlated with knowledge about ichthyosis in particular. So the take-home message is, if you're somebody who has a young child or someone who's already a teen or a young adult, get them educated. And particularly this was mostly even more so a problem in the younger set of this. They really knew very little about the disease. Now, I'll bet people who come to this meeting are a bit of an exception and not typical of what might have been in this survey. But nevertheless, that was very important, so I applaud all of you who do come to this meeting and get the teens knowledgeable. Now, stigma and ichthyosis we've been working on here. And I'm going to tell you that we just studied a group of almost 1,700 children using a new tool that for the first time measures ichthyosis in children and has what we call a proxy version, where the parent can also respond. And we tried to look at the impact of how much stigma there was and the impact of it, particularly on mental health, because stigma is something that can impact your anxiety levels, your depression, how your functioning is, your peer relationships. And stigma, just to tell you, can be external. So someone may see that you have scaly skin and actually take some action because of that makes you feel bad. But it can also be internalized. And I have to say, I don't know if Lowell's sitting in the audience, but when Lowell was talking yesterday, I kept thinking about internal stigma, or perceived stigma is the other name for this, where he was so worried about the scales on the floor that it totally consumed him. That's incredible concern about stigma that's not coming from anything outside that's happening, but rather is internal. So this is something that we want to measure, and this new score has been helping us to do this. And there were some patients, some individuals with ichthyosis in this group of almost 1,700, and I hope you saw the poster that was here. Are they still up, posters? Oh no, okay. Well, I hope you took a look at it. They've always stopped, so we'll keep going. We can break through the windows if we need to. So at any rate, basically it showed that in this group of children with chronic skin diseases, ichthyosis was one of the few at the top. So there's a lot of stigma, not going to surprise anybody here. But why did we do that study, and why do we want to know about this in ichthyosis? It's because this kind of quantitative data is what can resonate with regulators. It resonates with the FDA. It resonates with insurance companies that have to pay for your medication. It resonates with industry that we may need to convince to put an investment into studying new medications for ichthyosis. And it can also resonate with the public, this kind of data. So all around benefits to really learn about it, not to mention that if we study this, we find it may be something that we want to make part of our evaluation, and then be able to not just try to treat the ichthyosis, but try to treat the person as a whole. And that may mean getting some help with the sense of stigmatization or the anxiety or depression or other issues that it may lead to. So this was a study that we had here. I do want to tell you about some work that Andrea Rustad, a medical student I think many of you met, she worked with me on that book that many of you have seen, the Skin Invincible book. She's very interested in the bullying, and this has turned out to be a problem, of course, in ichthyosis. It's strongly correlated with poor peer relations and moderately strongly with stigma. But what we find is that the parent's perception can be quite different from that of the child. And the parent's perception, what we found, was correlated then with parental depression and anxiety. It almost didn't matter what the child was telling us in surveys about bullying or stigma. The parent's perception was what correlated with their degree of depression and anxiety, which makes sense. But nevertheless, it was the difference between the parent's perception of the child's bullying and what actually the child was reporting. Child's report didn't matter. It was the parent, but it also brought out that there was a fair amount of depression and anxiety. In addition, the reduction in family quality of life that we assessed was more closely linked to the parent's perception of bullying and that then of the ichthyosis severity than the child's. So the advice here is to be on the alert for bullying, educate teachers, principals, classmates about ichthyosis, and then counseling can really make a difference. And we should never be afraid to think about that and get children help. And, you know, bullying can occur for adults too, so just mention that in some kind of different ways. And most of the bullying that we saw also was at school. We hear a lot about cyberbullying, but it was in the presence that this actually almost always occurred. And I also would add that parents shouldn't be afraid to reach out for support. We all know when things are going on with our kids, it very much profoundly affects us as well. So what other studies are we doing here? One last reminder, raise your hand if you're part of my team. I see a bunch of people sitting back there. These are all ladies who can help you with surveys. We really want to get some more. We did have a survey here, which you can still do, which I don't have a QR code for, that looks at what I was just talking about. And you know what? We want to look not just at children, we really want to know what's happening with adults. So this year, for the first time, we created surveys that actually bring in data on adults. This was something that many of you had put down when we asked at the last meeting about what you wanted to see more on. We responded to that here. So please, if you're an adult and you did not do this, make sure you find one of these people. They'll all be at the front. They can, if you have a little bit of time before you jump to the airport, they're happy to do that. We had a question about joint pain because our biomarker work and experience suggests the risk is increased and the onset is earlier in life with joint issues. We really want to develop a tool for self-assessment. That means if we have a good tool for self-assessment, we can do more without having you come into the office. We can have you, using a validated tool, rate your own ichthyosis and follow that regularly. And that really opens the door for engaging more people who may not even live close to an academic center in doing research. So we want to develop this self-assessment tool. We also really wanted to look at the co-occurrence of allergic disorders. So it's hard to know. Everybody knows what eczema is. You all know somebody with eczema, but when you're covered with redness, scaling, and maybe you're itchy, how do we even know if you might also have some eczema? And so we're trying to dig a little bit more deeply into this. Some of our other research has shown that there may be a difference with people who have more allergies or asthma or high levels of something called IgE in your response to certain medications in terms of how it helps your disease or your itch. So we're trying to look at this a little bit more, and this was another study just trying to see if there are any signs of some of what we call atopic problems, asthma, hay fever, food allergies, et cetera. And then for Harlequin ichthyosis, we had a study here. I know there aren't lots and lots of people, but better characterizing the clinical features. Now I'm going to stop here for a minute. I have more to tell you. Anybody here not have a phone? Well, I want you to get out your phone if you have one, because if you here have not taken the 1 minute that it takes to do that self-reported severity tool or some of these other ones, please, this is the last chance for anything other than the stigma study. You can just do them by doing this QR code. Now, we're going to send something out later with this because we know not everybody made it to the conference. We want to hit more than 100 on these types of surveys, so we have some really meaningful results. So please, leaving it up here for a minute, take a picture. You can decide later to do it or not, although I would urge you, if we don't have the numbers, it's meaningless. And I consider this very low-hanging fruit because it's so easy to do, and if we can get the data back, we can have validated tools. We can have good responses to these. I'm just going to wait for a minute till they come up. We'll wait a little bit longer here. And I'm thanking all of you in advance for spending just a little time to do this. It's the kind of thing you can do sitting at the airport. All right, anybody need another second? Well, 1 or 2 more, okay. If you didn't get it, the team sitting back there, you saw their hands before, they have QR codes on paper. You can just walk over while I'm talking. Just get up and walk while I'm talking and you can do that. Okay. All right, so really quickly I'm going to go through a few other things and then pass the microphone on. At past meetings, we've talked a little bit about how exciting it was that we were able to do this biomarker work across the spectrum of ichthyosis. That consistently, and this has now been repeated by other investigators, there was a pattern of immune cell shifting towards what's called the Th17 immune system. That is, particular types of T lymphocytes that have a particular pattern that we call Th17. And that this is the same thing that we can see just as a response to microbes. Everybody knows that we've got microbes that are a bit shifted when we have ichthyosis skin, right? A little bit more of the Staph aureus, maybe a little bit less of certain things. I'll talk about that in a minute. Actually, I didn't talk about that. Len is here. Len's going to talk about that. Oh, he's not? Oh, I'm sorry, I took out the slide. Okay, well, I'll tell you then. With respect to the first one, the microbe studies have shown that there's more of something called Staph aureus, which you've probably heard about because it causes a lot of skin infections. There's also more of something called Corynebacterium, which we see go up as a body's response to try to kill off staph. But we saw a decrease in, for example, the bacteria that's associated with acne, or a common yeast that we have on our skin called Malassezia. And these are down because we have less what's called sebum, the oils that we see more of when you start to get acne. And I have to sometimes say that to people that I see with ichthyosis, well, one good thing, you probably won't have a big problem with acne, which is very true. So we did in fact see some microbial imbalances or things that are changed from healthy skin that we know can elicit this Th17 response. And I would say that probably across the board, one of the reasons we see this is because of that. But the big question that we wanted to ask, because we're always thinking about therapy, is does that Th17 excess drive inflammation, drive scaling, because that's exactly the same pattern that we see in a common skin problem called psoriasis. And then, if you take it a step further, recognizing that we have a lot of new, pretty exciting treatments for psoriasis that have been life-changing for individuals with moderate to severe disease, could we repurpose these? Could we use these towards treating? And I'm going to give you, in a nutshell, what we've found over the last few years. One is the first group that we tried it in was 2 people, as seen on the right, who had a rare type called erythrokeratodermia with cardiomyopathy. And in fact, the girl that you see on the left had already had a cardiac transplant. I don't think anybody in this room has this, but it happens. And when it happens, it can be very severe. This baby that you see on the left was already on the cardiac transplant list. You can see bright red, scaly skin. And in the children on the right, because we were able to look at one of them and do these biomarkers, verify this type of shift, we put them on this drug that's used for psoriasis. It's called ustekinumab, and it was life-changing. And this baby, after 1 month, was doing better, came off the cardiac transplant list, and baby in South Carolina who's still, to my knowledge, last time I checked, still on this drug doing well. So for this particular disease, it seems to be pretty consistently working. A disorder with a lot of redness. Secukinumab is another one of these medications, and in a somewhat related disorder called SAM syndrome that has this red scaly skin with inability to gain weight well, recurrent trips to the hospital because of infection. It has, everybody used, decreased the infections, helped with that weight gain, and then variably has helped with the skin. There were anecdotal reports, meaning a case report here, a case report there, maybe a few in a particular article, of using these kinds of agents for other forms. We saw it for congenital ichthyosiform erythroderma, and we saw it for Netherton syndrome. Here's just a patient of mine who, after just a few months, looked great. This is showing him at 8 months out. And at one point, we actually stopped it because we have to give him live immunizations, and that's what you're supposed to do. And he was just horrible off of it. Put him back on it. Did well. Many patients who are having that. Now, that said, these are anecdotal reports. And as scientists, we always want to ask the question, how do we know this is really working? And to do that, you do what's called a double-blind, randomized, vehicle-controlled trial. So we took across the spectrum of different kinds of ichthyosis, a group of about 20 people, and what we did is we randomized them. So for 16 weeks, they would either get this medicine or they'd just get some salt water. And what we found is that it didn't work for all forms of ichthyosis. We, by the way, kind of a natural history study in a bit, because we saw a lot of fluctuation, including getting better, including getting worse, in those who were on the placebo. Regardless, the best responses, and this is the punchline, were with CIE, and even more so with Netherton syndrome. Nobody with lamellar ichthyosis saw the kind of improvement that we were hoping for. There were some limited responses with epidermolytic ichthyosis, but it was the kind of thing where people were telling me they were feeling much better, they were using less medication, less moisturizer, these kinds of things. I didn't see a big difference. When we're looking for ways to treat, we're looking for big differences. We don't want little subtle ones, although of course people wanted to continue because subtle ones made a difference to them. But that's the bottom line for these. And so we're doing more work in this. We have a paper coming out with about 100 patients that were gathered from many sites that basically shows the same thing. This is a trial that we recently completed we had 12 children in this, 6 with Harlequin ichthyosis. So we thought that would be great. And although we did see improvement, people who wanted to stay on, not everybody, that we did find that it wasn't a dramatic improvement. So I don't think this is going to work for whatever reason with Harlequin ichthyosis. And I'll just put that out there. But then there have been some people who have seen more success. So some people do well. Some people don't respond, even with these types. We need more research in this, but I think I would restrict it to these types that had better improvement. I'm just listing some of the results that we saw up here. So for example, one patient with Netherton syndrome had a 71% reduction in the signs of redness and scaling. One with CIE had similarly dramatic life-altering changes. We don't know. We don't have biomarkers for this. So at this point, we're just continuing to try people on these kinds of medications. Now, I'm just going to put this up because there's a different pathway that's called the Th2 pathway, which is kind of the allergy pathway, which is a reason we're kind of interested in this. There have also been people who have gone on dupilumab. If you've ever heard of dupilumab, it's not that new anymore, a drug that's out for as young as 6 months of age with moderate to severe eczema. And for most people, it really is life-changing. It is an injection every 2 to 4 weeks, depending on age, but it's FDA approved, and the beauty of it is that we don't have to do labs or no side effects, except the patients with eczema have a low risk of getting some eye inflammation. So we happen to be doing a study right now. If anybody's interested in this, I know at least one person here specifically asked about itch. You have to have a fair amount of itch in association. We know that's highly variable. But talk to me about it, because there are airlines, for example, that might help to fly people out. There might be ways to do it if you have an interest. We monitor for 8 weeks, and then we get you on the medicine and compare it. And you can get it free for 2 years if you're part of this. So again, have to be itchy. Now, retinoids, most effective medication we've had, particularly for scaly forms of ichthyosis. Unfortunately, none of them yet have an approved indication. So the first potential topical retinoid specifically for ichthyosis, that is a topical isotretinoin, in earlier studies showed an excellent reduction in scaling. We have an ongoing trial, but it's really got data in. We're going to hear from Alan Mendelsohn in the next section. He's here from the company that's been moving it forward, but really maximal use study to look how much was getting into the blood was presented in a poster here. I hope you saw it. Really looks to be very safe from the standpoint of anything systemic, and showing some really good signs. So I don't have the official data yet, but this is showing the investigator global assessment improvement at 12 weeks that you can see from some of this research. And I think, as you can see, there were some people who did very well, some people didn't do quite as well, but in those who did well, I mean, look at the difference. It really can make a big difference. Now, we're using a topical retinoid in the form of tazarotene, but it's hard to get for some people. And it's not even approved until you have acne age, because it's for acne, and it's very expensive. So very excited about the possibility of getting something on label for ichthyosis. In fact, it would be the first on-label medication. So a big pharmaceutical company called Leo is moving that forward, and keep your eyes open for it. Now, what other studies or papers you may not be aware of? Some new directions. One, a big consortium in Europe is publishing some guidelines on care of ichthyosis. We'll make sure that that's available online for you, because it's the same guidelines for you as what they're doing in Europe. And in fact, I sat on that panel, and there were a few things there I said, we do it a little differently in the United States, so that's all been incorporated. Also, a consortium of 15 experts worldwide have been convening. Keith and I are both part of that. And we're looking, as I have just mentioned to a few of you, at reclassifying to think in a different way about ichthyoses, because what we're learning is that the best treatments are the ones that are based on the underlying mechanism of ichthyosis. There are some different ones, but for example, epidermolytic ichthyosis falls into the keratinopathies, and they're a whole group, including outside of what we consider to be part of FIRST, but we're going to learn across the board, because I think something that could work for one could work for another, and I'm going to mention that in a little bit for that. But then there are also, for example, with Netherton syndrome, the effect of proteases getting to be too high, or connexins, the communication that goes on across that group. Or we may be talking about all of the lipids, the ceramides in particular, that everybody who's born as a collodion baby, that's what's going on. Whether it turns out to be lamellar ichthyosis, whether it turns out to be CIE, with all the different subtypes, we have to start thinking pathways. We have to start thinking about how these are functioning to get to the redness, to get to the scaling, because that's going to be new treatment. So that's going to be coming out. We need to understand the natural history. There is a study coming up, natural history of Netherton syndrome, but we need to think about this across the many types of ichthyosis. As I mentioned, doing a study that had a double blind with a placebo-controlled. During the course of that, went up, went down. We have to understand, if you have epidermolytic ichthyosis, you know in the summer when it's hot, tends to be different from, for example, when it's cold weather. So there are these differences, and they've never really been effectively studied or chronicled. We need to do that. We need to understand why some types of ichthyosis respond and others do not to medications in trials or anecdotally, and that's one of the reasons that we're collecting skin. Sometimes we collect blood, particularly during studies. And we're looking at that because one of the things that we want to do, we don't want to just know about what's causing the disease. That's important. But also during trials, we want to collect these so we can, if we get large enough numbers over time, find patterns that will allow us to predict, especially if we're going towards some medications that may be expensive. I will mention there's a new study. I know that Beringer Ingelheim was here looking at Netherton syndrome that with an IL-36 receptor. You have no idea what that is. It's interleukin 36, but it's upstream in pathways that can lead to both Th2 that I mentioned for allergy and Th17 that I mentioned is really up across ichthyosis. So we're hoping that that looks good. It will be looked at for Netherton syndrome first. So now, speaking of Netherton syndrome, I'm also very excited about this pathogenesis approach. Gene changes in SPINK5 give you a deficiency in a protein that really activates what are called kallikreins that break down the barrier and also lead to skin inflammation. And so studies, we had Coin here, there's another one called Sixtera that's done studies in Europe, and more coming. Those are with topical kallikrein inhibitors. There are going to be some coming out that are going to be systemic. There's also going to be something that adds back the LECT protein. And there are other medications in development talking about this all along this particular pathway, as I've shown here. Now, you may have heard about EB, epidermolysis bullosa blistering disorder, and the really hot information about a new topical gene therapy for this. Now, they started with that because there's no barrier when you have blisters and you have open wounds, so it becomes very easy to get everything through. Even though we have a barrier defect with ichthyosis, it's not clear that this delivery system that has these big genes in it is going to get through, but this is next coming to lamellar ichthyosis. I suspect that in another year, within the next year, trials will be starting for that, so keep your eyes open on that. I also want to say a little bit about epidermolytic ichthyosis because there's some new directions there. These are all keratinopathies, and there are several of them. And what happens here is that the instability of the keratin network, because the protein is there, but it's not functioning right, leads to a collapse of the network, and that leads to blistering. It leads to aggregation of these little keratin filaments that should be nice and spread out. It leads to cell stress and inflammation. So in trials for EB simplex, which also is a keratinopathy, there was a product called Tolisure, topically applied, that selectively cleared the mutant keratin 5 and 14 and these keratin aggregates to strengthen the keratin network and reduced blisters and inflammation. This will be moving next to bigger trials, but also to epidermolytic ichthyosis. So we're going to see that moving into our network. And we've had representatives here as well from the company Biomendix that makes this. Other potential directions can be inhibiting inflammation because that's increased, and there are some potential ways to do that. And then inhibiting epidermal growth factor receptor, because that is something that's very important in regulating whether cells grow or whether they normally mature. We see very high levels expressed in ichthyosis, at least the ones we've looked at so far, certainly in terms of the keratinopathies, and we want to look beyond that to see if that will help beyond that. Part of what we're thinking about for the future is a trial of an oral medicine that inhibits epidermal growth factor receptor action called erlotinib. We're hoping to have a trial of that in the future. We're applying for some NIH grant now to do that. Finally, I would just say, if you want to keep in touch with what's going on with trials, check out clinicaltrials.gov. Go on and just put in ichthyosis, and it'll show you everything that's happening. Everyone can access this. So in conclusion, there's lots going on. Fill out questionnaires, please. We're happy to answer any questions for you about trials. Be on the lookout through the FIRST website or on clinicaltrials.gov to find something. And many of these studies have funding or can try to help to get you there so that you can participate even if you're from afar, or may involve just questionnaires or telemedicine. Now, I took a bit of time. I'm going to pass this on to Keith, who's going to tell you about the registry.
So I won't be talking about any of the specific projects that we've done related to quality of life, depression, anxiety, or any of our other work in therapeutics, and focus my discussion entirely on the registry. I also want to give some time to a couple of the extraordinary investigators who presented posters today who are going to do quick shots for us. So the registry started a number of years ago. We've now enrolled 1,700 patients. We've been able to provide genetic diagnoses to 1,450. We do pre-screening for mutation in 72 genes. We identify mutations in about 85% of subjects. And we refer the results of this to referring MDs when the results are available. We provide a pathway to CLIA-certified confirmation. These are the genes that we find that are mutated when we look, and many of you see genes that you know and love, but there are about 13% of patients that have mutations in 36 other genes. We've discovered a lot of genes via the registry, about 15 in total. We have 3 new novel genetic causes of ichthyosis that are in preparation for publication, and the registry has resulted in 43 publications, which I'm not going to detail today. We did recently publish a genomic and phenotypic landscape of ichthyosis, analyzing the first 1,000 kindreds. And this is really important for diagnosis because we're able to identify 266 novel pathogenic variants in 32 genes, which is really important for transforming our capacity to do clinical diagnosis. It also expands phenotypes due to mutations in known genes. There are a lot of different clinical tools that have come from the registry that include collaborative projects with Dr. Paller and many of the people that you've seen here at the meeting, including our Medical Scientific Advisory Board, including the Visual Index for Ichthyosis Severity, which really powered the TIMBR-LEO trial, and now the ISS, which I think is a more modern version. We've done a bunch of work with trials, and so the registry is one of the primary ways that you can be connected with new trials that are specific to your genotype. And so we have completed enrollment for a number of different trials and have inquiries from a number of others. But I think that what I'm most excited about isn't the work that we've done before, but it's really the work that's to come in the future. And much of that is going to be powered by what we saw today. So as we've discussed, the majority of therapeutics that we've talked about don't work for everyone, and they don't even work consistently within individual genotypes. Patients can respond initially, lose response, use a different drug, lose response, and be treated with yet another drug and lose response. And so what we want to understand is, at its heart, what specifically is going wrong when you have a genetic mutation in every single cell within the skin. And so shown here is a diagram of the skin going from the underlying fat in the vasculature with all of the immune cells moving into the dermis, which is the support of the skin, and then our keratinocytes, which undergo this regular pattern of differentiation where they need to create structural stability but also elaborate lipid to create the barrier. And so we've done very well at identifying individual mutations, but what we haven't done terribly well is focus less on the differences between everyone in this room, but instead thinking about the similarities, because everyone has scaling, everyone has erythema. We don't fully understand why some individuals are immune activated, and so what we can do is we can now take those skin biopsies that many of you provided to us and do a variety of procedures where we can look at each of these individual cell types, whether it's a blood vessel or an immune cell or a nerve, or any sort of other cell that's contained within the epidermis, and figure out what is that gene doing wrong at a specific developmental stage within the skin. And I think that that is likely going to be the equivalent of the fact that we have, one gene that one gene that we target for high cholesterol, and that's HMG-CoA reductase, and we've essentially centered many different drugs around that one pathway for statins. And so we really need to be able to think about what are the genes and what are the pathways that are common to all types of ichthyosis that we might be able to target so that we come up with highly efficacious therapies across multiple different genotypes. And so this is going to be enabled by a lot of advances that have happened in the world of being able to understand gene expression and the proteins that are present within the skin. I will highlight one really important study, which I hope you'll also scan today. So many of you recognize that ichthyosis is something that really uniquely affects children in terms of their growth and development. And yet we haven't been able to systematically classify that. And so we've been running a long-term study to assess growth and development among children, and essentially affected individuals and their unaffected siblings less than 30 years old are encouraged to apply. What we'll do is we'll obtain your growth charts and do comparisons between the affected individual and their unaffected sibling, and also to the general population, so that we can prove conclusively which genotypes have the largest effect on growth and development. And this is really important for thinking about things like access to services and also changing the way that we think about how pediatricians and pediatric dermatologists treat children with ichthyosis. And now I'm going to turn the floor over to some research quick shots, and we're going to start with Stefanos.
Thank you, Dr. Choate. That was really great. As Dr. Choate mentioned, my name is Stefanos Katsoukos, and I'm a PhD candidate at the University of Colorado Anschutz Medical Campus. And I'm really excited to share some data from the lab today regarding ex vivo genome editing of one particular mutation in one particular gene that causes autosomal dominant lamellar ichthyosis. Do we have the clicker? Oh, sorry. Gotcha. Thanks. So many of you have probably heard of CRISPR-Cas9, a really revolutionary editing or molecular biology tool that allows for the precise modification of DNA sequences, including disease-causing pathogenic variants. In fact, including many of those pathogenic variants that are causal in all of the ichthyosis that we are sitting here with today. And so in the lab, we were able to apply CRISPR-Cas9 genome editing to one particular genotype of ichthyosis that is causal in autosomal dominant lamellar ichthyosis, and that gene being ASPRV1. In fact, we particularly targeted one pathogenic variant within that gene, of which there are quite a few, that being a 595A to G mutation. So we were able to source fibroblasts from a kindred that harbors this particular variant. By the way, this particular condition only being characterized back in 2020 by, in fact, Dr. Choate and Dr. Paller, and designed a CRISPR-Cas9 genome editing system in order to correct that pathogenic variant and really revert that G back to an A. And in that graph I show you on the right, we in fact see we see that correction. Before the application of our CRISPR-Cas9 genome editing system, we don't see any correction of the pathogenic allele. But after we subject our fibroblasts to CRISPR-Cas9, we do in fact see a reversion of that pathogenic G back to an A, shown here in green. So this really provides a basis for developing CRISPR-Cas9 genome editing, not just in ASPRV1, which is in fact a particularly rare subtype and genotype of ichthyosis, but all different subtypes of ichthyosis, including TGM1, ABCA12, and we can go down the list. I'm really excited about this work, and I'm excited to continue it in the lab, and I'm really appreciative of the research update today, giving me the opportunity to share this work. Thank you all.
Hi everyone, I'm Hannah. I'm part of Dr. Paller's team. And today I'll be sharing some results from some of your tape strips and skin biopsies that you've been sharing with us. So, your skin is made up of the top layer called the stratum corneum. Sorry for the boxes, they got a little distorted on the big screen. And your stratum corneum has cells called keratinocytes. And these keratinocytes are tightly packed, but between these cells, there's a bunch of lipids, which are these fats that can be made, which ceramides are a type of. And these ceramides are made up of sphingosine bases and long-chain fatty acids. And so some studies have been done to show that there are decreases in long-chain fatty acids in skin with ichthyosis, but no study has yet shown that there are any changes in ceramides or sphingosine bases. So what we did is with your skin samples, we wanted to quantify the difference in healthy skin versus ichthyosis skin in changes in ceramides and sphingosine bases. And that's really important because we want to be able to see what is decreased so that we'll be able to replace that, or on the other hand, what increases in ceramides are actually damaging your skin. So what did we find? We found that in every type of ichthyosis, there's markedly reduced ceramides. And so the next question we have to ask is, why do we see this decrease? So what we also did is this method called RNA sequencing, and you can actually see what genes are reduced or increased in expression that are involved in this ceramide synthesis pathway. And so we did see some reduced or altered expression in those genes. And so the next thing that we did is we also looked at kind of how does this decrease in ceramides correlate with disease severity. So we also were able to see that was correlated with an increase in water loss in the skin barrier, increase in scaling, and increase in erythema. So what the next steps are really for this is we really need to continue with your samples. So when you are donating your samples through tape strips and biopsies, it really helps us. And so we urge you to continue being involved in research in that way. Thank you.
Thanks. Hi, my name is Ryland. I'm an MD PhD student in Dr. Choate's lab. So given the interest in inflammation and ichthyosis and potentially repurposing immune-based therapies that Dr. Paller mentioned earlier, we wanted to use a new method which allows us to look at inflammatory markers directly in the skin from patient samples. So we use this method called RISH to look at 10 different druggable inflammatory markers in 7 cases of ichthyosis with confetti, which is a specific type of ichthyosis. What we found is that we did not see the cytokine IL-17. We did see a big increase in this cytokine called IL-36, which actually Dr. Paller did mention earlier, can contribute to this Th17 inflammation that was mentioned. This cytokine was specific to the affected skin and at much lower levels in the unaffected skin. And we then looked at a lot of cases of ichthyosis due to different genetic variants and compared the levels to inflammatory skin diseases, psoriasis and atopic dermatitis. And we found that a handful of cases, a little under half, have levels similar to inflammatory skin disease. And so we're looking into whether this measure might be something that could potentially predict response to immune-based therapies. So thank you for listening to the research updates and for contributing to our research this year.
Okay, we're going to move on to the next session, and I don't know where Chris is. In the back? Okay, well, do you want to go ahead and introduce that? Could we also have people who maybe spoke in this sitting up for at some point so people could ask questions? Because I bet there are some. Okay.
Absolutely. We're like right on schedule right now. Are there any questions before we go into the clinical trials discussion? All right. If you have them as we go along, we'll address them. Let's pull some chairs up and create like our panel. Yeah, I think Lauren. I'm missing. Where's Lauren? Oh, there you are. Okay. Come on down. Come on up. Go ahead. Take a seat.
In the meantime, it was just really exciting research from some young people who were involved. Just want to make sure there weren't any questions for them while we're assembling. I'm sure you're pretty excited by some of that. Yes, go ahead, Denise.
So the gene editing, is that looking at a cure, first of all? And are we looking at a future where we're editing genes for babies before they're born, or editing genes for adults that already exist?
Yeah, I guess I can take that. Thanks, Chris. I think there are a lot of unanswered questions in that realm, and one of the questions that really keeps me up at night as I give you that update is how do we deliver these systems into the skin, and not only the skin, but do they localize or do they get to the right layer of the skin to actually make a difference? Or it's not even just make a difference, but are they actually able to edit at an efficiency that would make a difference? But what really gives me hope in particular is that, especially with recent FDA guidance and really grouping platform technologies together for a lot of different indications within similar diseases such as ichthyosis, I could see maybe a day where when we figure out that delivery problem, we could really target a lot of different genotypes together with a similar product and switch in and out the bits and pieces that target genes specifically and genetic variants specifically. We're very far, and there's a lot of work to do in the lab, but I'm really excited about the work.
Can you address, that's
Okay.
Can you address the efficiency of this? You mentioned that, you see it going up to .08. What does that mean? And I know you study this a lot, so how is that moving forward to become more efficient so that if you're exposing cells to it, most of them are actually having that happen?
As Dr. Pollard mentioned, the efficiency of my system upon first pass was 8%. That to some degree means that for every 100 cells that were hit with CRISPR-Cas9 genome editing, only 8 actually showed a correction of the pathogenic allele. Now, that's, in the world of genome editing, that's actually really great efficiency, but in the world of does that actually would that make a clinical difference, in the skin of us affected by ichthyosis? We don't know that. But, one approach we have in the lab are what's called xenografts, where you can actually take human skin cells, fibroblasts and keratinocytes, graft them onto mice, and actually they form full human skin equivalents with the correct dermis and epidermis. And one thing that, one approach that I'm thinking that could answer that question is what proportion of corrected cells do we need in the epidermis and dermis to actually make a phenotypic difference and then ultimately a clinical difference? Those are again some unanswered questions, and there's always improvement that we can be making in genome editing and very much ongoing efforts in the lab. Thanks.
Thanks, Stefanos. All right, we're going to move on to the next phase where we're talking about participating in clinical trials.